My name is Mani, and I'm currently a PhD candidate and also a young researcher in the Faculty of Science and Engineering from the University of Nottingham, Malaysia. So my research investigated the effect of polyethylene terephthalate microplastics on rat isolated tissue function and liver enzyme. My area of interest is because I wanted to gain insight into the potential health risks that today youth and generation face. So, plastic microplastics pollution has emerged as a threat to not only the environment, but also the human health with far-reaching implications for the well-being of the future generations. So, according to Plastic Europe 2024, they published a fact about the plastic production in 2023. In 2023 alone, there is a total amount of 413.8 million tons of plastics produced, which is a lot. And among that 400 million tons, 6.2% of it is polyethylene terephthalate, PET. I believe that all of us here has used products that is made of PET before because many consumer products packaging is actually made from PET, including the detergent bottle, plastic drinking water bottle, takeaway boxes, and whatever plastic product that's labeled as number 1. So all these plastic products, they will degrade into tiny, tiny pieces called microplastics. And microplastics can remain in the environment for a very, very long period of time. For example, in the aquatic environment, in the freshwater, marine water, marine sediments, in the air, and of course in the soil. So as I mentioned earlier, it can invade the water system and is ingested by aquatic organisms. For example, fish, shellfish, and eventually all these microplastics will enter the body of living organism, including human. So what I wanted to find out is what happened when PET microplastics is exposed to animal tissue and liver enzymes so that we know that if the same situation happened in human, what are the possible consequences? So this is the aim and objective of my research. My main aim is to investigate the effects of PET microplastics grinded from plastic drinking water bottle on rat isolated tissue, specifically the blood vessel of aorta, the airway, trachea, and also the bladder, and also to see what is the effect of PET microplastics on liver enzyme activity, specifically the CYP450 enzyme, which is an enzyme that preoccupy in the liver, specifically these 3 isotypes. So how do I carry out my experiment? First, I will grind plastic drinking water bottle labeled as number 1 into microplastics into powdered form, and I was subjected them into multiple characterization tests with various analytical technique such as FESEM, EDX, and FTIR. And next, I will incubate these microplastics with the 3 types of isozymes that I mentioned earlier using the screening kit bought from Live Technologies. On the other hand, the Sprague Dolly rats, which are the white rats, will be purchased from University of Kebangsaan Malaysia Animal House, and they will be sacrificed on the day of the experiment, and the tissue will be excised and cut into segments or strips, depends on the tissue type. And the tissue will be incubated for 22 to 24 hours with microplastics prior to the organ bath experiment. So on the right-hand side of the screen, this is an example of the organ bath experiment setup. So if you can see my mouse, not sure, but you can see that the tissue will be either hooked or tied to a metal hook and connected with a surgical string all the way out to the force transducer. So this force transducer can actually detect any changes in the tension. For example, if the tissue contract or relax, then we can detect the signal, and then the tension or the signal will be captured by a data acquisition system and further showed in a laptop. So why I wanted to grind the microplastics from drinking water bottle, because I want to make sure that they are actually made of PET. So by assessing the FTIR spectrum, I confirmed that yes, this drinking water bottle, our one-time drinking water bottle, is actually made of PET. So I compared the FTIR spectrum with online databases called OpenSpeci, and it confirms that the drinking water bottle are indeed made of PET. Next, I subjected them under a microscope called a field emission scanning electron microscope, FESEM, and we can see that the morphology of this microplastics particle were found to be in the forms of fibers and fragments. Some of them has longer stretch, which are fibers, and then some fragments type. So the average length and width of this microplastics particle are 85 micron and 21 micron respectively, and they are actually corresponded to the size of microplastics found in human body, such as in the blood, in the liver, and also in the lung. So after I incubate the microplastics with all different type of tissues, these are the results that I obtained. So we actually find out that it actually causes an enhance in the relaxation of the aorta. So if you can see at the 1 mg per ml concentration group, the control tissue actually relax lesser than the test group. However, it is interesting to note that there is no significant changes with 0.3, the lowest concentration group, and the higher concentration group. So we also tested it on the aorta contraction, but there is no changes in the aorta contraction. The second tissue that we tested are trachea. So just like what we see in aorta, it actually shows reduced contraction, but it also happened only in the lower concentration group. So in the first two graphs at the top of the slide, you can see that actually the control tissue contract more than the tested group. So this is interesting because if the trachea has a reduced contraction, it can lead to accumulation of mucus in the body, and it will increase the respiratory infection risk. However, when we tested on the trachea relaxation, there is no significant difference. And move on to the last tissue that I tested. On the bladder contraction, similarly to the trachea contraction, it shows us reduce in the magnitude of contraction, but similarly, at the higher concentration, there is no significant effect. So we found out that the effect of microplastics is only observed in the lower concentration groups throughout the 3 different types of tissue. We speculate that this is because the microplastics can disperse better and also interact better with the tissue, and a lower concentration of PET microplastics is less likely to aggregate, so allowing more effective contact. And so we conclude that microplastics likely exert its effect through a physical interaction, and this is not dose dependent. So now that I've seen effects on the tissue level, I wanted to move one level down into the molecular level. That's why I carry out the experiment to see the effect of PET microplastics on CYP enzymes, specifically CYP2C9, CYP2D6, and CYP3A4. So what is this CYP enzyme? CYP450 enzyme is a superfamily of a group of protein that are predominantly occupies the liver. And this protein is actually very important because 6 out of 57 of these isoforms to date are responsible for 90% of drug metabolism. So imagine if this is compromised, means that our body wouldn't be able to metabolize drug as efficient. So for these 3 enzymes that I looked at, it's interesting because only 2 isozymes show significant inhibition, whereas on the CYP2D6, it doesn't show any inhibition. So we speculate that this is because there are properties of their active site. So active site is like a place that substances can come in to interact with the enzyme and causes further subsequent action. So for CYP3A4, which has the most significant inhibition, it actually has a large and flexible active site that accommodate wide range of substrate, wide range of substances to make it more susceptible for interference with PET microplastics. However, with CYP2C9 that show moderate inhibition, it has a more restricted active site compared to CYP3A4. So it is less prone to interaction with microplastics at a lower concentration. And lastly, for CYP2D6, it remained unaffected. So the active site of the CYP2D6 is actually very distinct, and it has a very distinct architecture and very specific substrate specificity. So from there, we conclude that PT microplastics exert isozyme-specific effect rather than a generalized inhibition across all CYP enzymes. So to conclude my result, we found out that the acute exposure of PT microplastic to tissue did not impact the aorta contraction, trachea relaxation, and bladder relaxation. However, it changes the tissue function. For example, it increased aorta relaxation, reduced trachea contraction, and reduced bladder contraction. And on the other hand, with my enzyme study, it showed that microplastics selectively inhibited some of the activity, but it left other isozymes activity unaffected. And this difference may be related to steric hindrance effect, binding at other sites, or the properties of the active site that I mentioned earlier. So all this disruption could worsen disease in vulnerable patients by impairing normal tissue function and disrupting the detoxifying system in the liver. So what is the next step from my research now? For more lab work, we can investigate the impact of microplastics exposure on tissue structure by examining their histology, their tissue structure. And also we can have some mechanistic study to explore exactly how these microplastics interact and affect tissue function. So as the younger generation, all of us here, we will be the generations that are affected by this plastics pandemic. And hopefully through my research, I can contribute towards global goals and turn lab research into knowledge that communities and policy makers can act on. Thank you.